Result Life Science Academy. We are the institute based on Coimbatore. We are successfully completed 3 batches in giving coaching and offering coaching for CSIR-UGC nets. Our students are being placed in as a JRF and also the SRF, and in some candidates has been get selected as a lecturers in the Tamil Nadu. So we are completely offering an relevantly very high mode of coaching for the CSR students, and currently we are going for the next stage in providing a free online coaching for the CSR UGC net students. And not only we are giving this training for the CSR UGC net, and we are also offering training for the GATE, ICMR, DBT coaching also for the students who are going for for the postgraduate, like when the students who are going for interesting in appear for the IIT JAM. Also we are offering training for the students at low fees. We are been using an experienced faculties to train all our students. So this free online coaching it has been made for the CSR students who are not having enough time to go for the classes, regular classes. So this is the classes mainly we will be concentrating like on the important topics in the CSIR. When you're going for taking about the syllabus of the CSIR, it's having 13 topics. In the 13 topics, we are going for focusing upon the important topics like 7 topics, and we will be regularly updating the new classes in the online. And whatever doubts you are having over that, and we are giving a blogspot address at the end of the slides, so you can go over there and you can have any comments over it, and you can paste on that. We will be get back with you within 1 or 2 days. And today when you're going for today classes is mainly about the first one, that is molecular relevant to biology. In this molecular relevant to biology, we are going for discuss about the metabolism of nucleotides. And before we are going for studying about the nucleotides, we will be very much known about what is meant by nucleotides. So when you're going for checking out the nucleotides in our humans, we are having two kinds of nucleotides. Other one is purine bases, and next one is pyrimidine bases. And before we are going, purine is nothing but is a 9-member ring, and this is a pyrimidine is a 6-member ring. These are the informations already we have made very much familiar about this one. And in case of the purine bases, we are having before nucleoside, we are having another one term, that is nothing but nucleoside. So nucleoside is nothing but sugar. All we know about that the DNA is made up of 3 important molecules. One is purine or pyrimidine bases that will be get combined with that of the ribose, and we will be having one phosphate molecule. And there are 2 kinds of things materials are made up of in the DNA. One is nucleoside, and next one is nothing but nucleotide. So in case of the nucleoside, that is a new nitrogenous base along with the ribose, we will be calling as a nucleoside. In case of the nucleotide, that is nitrogenous base plus ribose plus phosphate molecule. This the backbone of the DNA is phosphodiester bond. So in coming classes, when you're going for discussing about the DNA, I will be explain clearly about how the structure of the DNA has been get made. So initially today we will be just look after what is meant by nucleoside and what is meant by nucleotide. And when you're going for the next one, so in this already we know that is a deoxy, that is ribose. What is a sugar molecule which is present in the DNA is deoxyribose molecule. In this you can, when you're going for this is nothing but how the nucleoside will be there. This is a nucleoside, and this is nothing but the nucleotide. When you're going for giving the structure itself, we can see this is the nitrogenous base, and that will be get combined with the ribose molecule. So we are telling this is a nucleoside. And in this combine, when you come to the next one, this is a ribose, this is a purine, next is a ribose molecule, and you can see this red mark is indicating that the phosphate, and all together this can be called as a deoxyadenosine 5 triphosphate. So this is nothing but it's a nucleotide. And purines, how the purines will be, what are the purines are present. So when you're going for checking out the purines in our body, we are having 2 kinds of purines are there. One is adenine, and next one is guanine. In case of the pyrimidines, so we are having 3 bases. One is thiamine, uracil, and cytosine. So we have been very keen, that is cytosine and thiamine is only present in the DNA, and uracil is present only in that of the RNA. That is the thiamine is get replaced by uracil in case of the RNA. When you're going for the structure of the nucleotides, so how we can see this bonds like the purines or pyrimidine bases that is going to be get joined with the ribose or deoxyribose. We have given the common structure of the nucleotides both in the DNA and RNA. In this, what kind of a bond it is formed between that is purine and also with the ribose molecule is an glycosyl bond. So just to key in, that is there are two kinds of bonds will be normally present in the things. Commonly we can see that is one is the glucosidic bonds and next one is the glycosyl bond. That is glucosidic bonds are formed between the sugar molecules. So when the sugar molecules is going to be get form a bond with a non-sugar molecules, it may be a lipid or it may be a protein. When that kind of the bonds will be called as commonly we can tell this as glycosyl bonds. So when you're going for the structure of this ribose molecule, we can see in case of the ribose molecule that both the 2' end and 3' end will be having the hydroxy group. In case of deoxyribose, in this 2' end won't be having the hydroxy group, it will be having the hydrogen group. And first one in the section 1, first we will check it out with the degradation of the nucleic acid, how the commonly the nucleic acid will be get degraded. So what are the proteins we are being get ingested to our diet, that will be goes into that of the stomach. The stomach, in case of the stomach, has been having the, already we might be familiar that it will be having an acidic pH, and also all the proteolytic enzymes are present in the stomach. So these proteolytic enzyme along with the gastric acid digesting the incoming nucleoprotein into nucleic acids and also the protein. These nucleic acids has been get acted by the enzyme called endonuclease. This endonuclease are nothing but we are having a nucleases is terms that is indicating that this is going to get degraded the nucleic acids. So there are two kinds of nucleic acids are there, RNA and DNA. So in this RNase is indicating that the nucleic acids, that is enzymes, are going to be get act on the RNase and breaking down into small branches. In case the DNAs that will be get acting on the DNA and break into small ends. So endonuclease are nothing but, what is the difference between endonuclease and exonuclease are nothing but exonuclease will be get removing the nucleotide from one end, and in case of endonucleases it will be get acting at the internal ends. So as a result of the action of the endonuclease, you will be getting some fragments after the action of the endonuclease. So from the nucleic acid will be by the action of the endonuclease will be getting the nucleotide. This further act by the enzyme called nucleotidase. So that is converted into two parts, that is nucleotide is broken down into nucleoside and phosphate, and nucleoside is again act by the enzyme called nucleosidase. So this will be again converted into base and ribose, where the starting product has been get formed. And what is the significance of the nucleotides? Why we want to discuss about this nucleotides now? Because the nucleotides are playing a major role, because this is a precursors for the formation of DNA and RNA synthesis. And next one, these are acting as an carrier molecules for chemical energy, like especially the ATP. So ATPs are commonly involved as an energy sources. In some cases we can find GTP, and in for the activation we require UTP. And next, these are the components of the cofactors like NAD, FAD and coenzyme E. And finally, these are the formation of activated intermediates, like as I said earlier, that is activation of the glucose molecules. We require this one. Kindly note this one. In case of the glucose to be get activated, we have to be get combined with that of the UTP molecule. Similarly, in case of the diacylglycerol, you can check it out. That is, it used to be get combined with that of the CTP, that is cytidine diphosphate. And next, this is more we will be known about that CAMP and GMP are nothing but the secondary messengers that is commonly involved in that of the signal transduction. Whenever we are having this in the CAMP and GMP, already we can see in the as a common secondary messenger in the G protein coupled receptors. And section 2 we will be coming to the topic like we will be discuss about the synthesis of purine nucleotides. So there are two pathways are there for the nucleotide synthesis. First one is a de novo pathway, and next one is salvage pathway. So for the synthesis of the nucleotides, in case of the de novo pathway, it will begin with that of the ribose 5-phosphate, carbon dioxide, amino acids and 1 carbon units. In case of the salvage pathway, the synthesis of the nucleotides is done by the recycling the free bases released from the nucleic acid breakdown. As I said, we have already seen on the degradation of the nucleic acid sections. In case of the first, we will discuss about this de novo synthesis. In the site of the action where this de novo synthesis will be taken place is, it is taking place in the cytosol, that is cytoplasm of liver, small intestine and thymus gland. And next one is, what is the characteristics of the de novo synthesis, that is purines, where the purines are being synthesized by the de novo synthesis. We can check it out like the purine synthesis done by the de novo one. In case of the salvage pathway, both the purines and pyrimidines will be get synthesized from that of the salvage pathway. In case of the, now we are discussing about the purines. The purines, where this starting material is 5-phosphorybases, the starting material, and PRPP, that is nothing but 5-phosphoribosal 1-pyrophosphate, is an active donor of that is ribose 5-phosphate. And AMP and GMP are synthesized. Where will be that product will be get end over here, and case of the end of the de novo synthesis is IMP. From that IMP only will be synthesizing the AMP and GMP. So before we are going for thing, this diagram we kindly go through that one and memorize these things. Already so many questions has been raised in the CSIR regarding the source for the derived for the particular carbon. When you check it out, the carbon dioxide is acting as a source for the one carbon molecule, and glycine is acting as an amino donor. Not only the amino donor, it is also acting as a donor of the carbon skeleton, and aspartate and N10 formyl tetrahydride, it's donating the carbon sources, and also the amide of glutamate, it is also acting as a source for the nitrogen. So you kindly check it out this one. So many times the question has been raised over here, and you will be very much keen about the carbon and also nitrogen sources. And tetrahydrofolate, it is one of the main important enzyme, important molecule, that is chemical moiety. So what is the main function of the tetrahydrofolate is, it will be donating the methyl group to that of the molecules. So for that we using the enzyme called dihydrofolate reductase. This dihydrofolate reductase is a common target for the cancer drugs. So next we will move to the section how the synthesis of inosine monophosphate will be taken place, because already we have said earlier that is purine biosynthesis is commonly end in that of the inosine monophosphate. From the inosine monophosphate we are going for synthesizing the AMP and GMP, because we are having only two bases, that is adenine and guanine. So where this will be get start, I said earlier, this will be get start from that of the ribose 5-phosphate. Kindly just check it out how many steps are there involved in the synthesis of inosine monophosphate is 11 steps. Where this will be get primarily takes place is, this will be takes place in the liver. So purine nucleotide biosynthesis. So what are the precursors? Kindly just note it down. What are the precursors that occur for this nucleotide biosynthesis? We occur 5 moles of ATP, 2 moles of glutamine, and 1 mole of glycine, and 1 mole of carbon dioxide, 1 mole of aspartate, and 2 moles of formate. Already I said earlier. So this is an important question. Already it has been asked in the CSIR, GATE and ICMR like questions. And another thing, you just note it down, what is meant by hypoxanthine. The purine basis without the attached ribose moiety is hypoxanthine. This hypoxanthine is playing a major role in purine metabolism. Thus we can discuss it earlier. As that is purine basis has been built upon by the ribose by several amidotransferase and transformation reactions. Next, when you are going for checking out the reactions involved in it, it is start from that of the PRPP, that is 5-phosphoribosal pyrophosphate, and it will be get ended into that of the IMP, that is inosine monophosphate. Kindly check it out. These the first one, the reactions 2, 3 and 5 are controlled by the gene called GART gene, and next one 6 and 7, which has been catalyzed by the enzyme called PAICS gene, and last two reactions has been catalyzed by that of the ATIC gene, that is aminoimidazole 4-carboxyamide ribonucleotide formyl transferase. And for that is the rate limiting step is first one is the conversion of that is PRPP to that of the PRA, that is catalyzed by that of the enzyme, that is GART gene, that is a rate limiting step. So kindly just note down, in all the metabolic reaction we have been, the examiners will be very much keen in about the regulatory points. So whenever you are going studying in the biochemistry part, you just check out the regulatory enzyme steps. And for thank you, and in next classes we discuss about the catabolism of the purine nucleotides. Thank you very much. For more information, kindly just log on to resolvebiotechno.blogspot.in. You can find regularly, we can find an updates of the bio classes in the CSIR-UGC. For more information, our office is located in the 316 Sridevi Towers, 5th Street Extension, Gandhipuram, Coimbatore, and our phone number 81484 52460. Thank you very much.